agents. So we have a policy that all samples that come to our lab must be in DNA RNA sheet, just to make sure everything is good. So you'll proceed to pick your sample. So just give you randomly your samples. Then we proceed to do the assays. If you have any question, you ask. So, but basically for extractions, we normally follow the protocol. It's nothing really complicated, but yeah. So every kit comes with a protocol. This is the protocol for that assay, for the extraction assays. So I'll just tell you the type, the sample type, then you pick the protocol that you use for this, for the extraction of that sample. Okay, I think I should have two. I don't know how you'll work because you only have one hood. You can take turns, Okay. Yeah, so I don't know if you need, do you also need to take you around or you know the instruments, all of them? You're familiar with some. So we have this, basically this is an extraction room. So you can use the kit to do extractions, but also we have the automated extractors, just like the one you saw on the other side. Yes, so you take turns. I'll just, you pick a sample, then we start to extract the samples. If you have any questions, you can ask. Okay. Yeah. So the first person can proceed to sit at the bench. Yeah.
I pick any.
So you pick any. Then I'll indicate somewhere. So I'll be able to judge your skills, pipetting skills at the end of it.
Okay.
Yeah. Yes, so you take a protocol. Give you a protocol. So the sample is blood in DNA RNA sheet. Yeah. So you pick which protocol works best for that sample.
I think it's no, this is blood. So page number. Really?
So these are human. These are just human samples, human blood samples, non-infectious. Yeah.
This is just. I think
it should be. Yeah. So these are the protocols.
Yeah, yeah. That's right. So I'm supposed to add. So I believe this is already 200 micrograms.
So this we have 100 of the sample, then 300 of the DNA RNA shield. Yeah.
So I'm supposed to add this.
So you're supposed to add. So you picked that protocol for you.
Yeah.
Okay.
Is it okay protocol?
No, it's not.
Okay. Why?
So we'll follow the DNA RNA shield protocol for this.
Okay. What's the difference?
So the difference is you'll see on the reagents that you use for the assay are different. So for these we had the biofluid cell buffer, which is red, but for these assay we don't have that. Yeah, so we follow the one. So normally you follow the protocol that matches your sample. Yeah, that suits your sample.
Okay.
Yes. So these are samples that are stored in DNA RNA sheet. Yeah. So you follow the protocol that says DNA RNA sheet.
And for this one?
Yes. So you do the biological fluids because it's a biological fluid, and yeah, it's in DNA RNA sheet. Yes.
So we are supposed to start with protein S.
Yeah. So you add 20 of protein K. Okay.
I think these are 20.
Oh, okay. So the total volume is found in microliter. The total. So we have 100 of the sample and 300 of the DNA RNA shield. Yes. Okay. Okay. So how much are we picking? How much of the protein is here? 20. So you go for the one that is closest to the volume. So that's 20. Yeah, that's fine. Okay, so the tip should be there. The small ones here. Just a minute.
All you need is to set your budget to 20.
This one, there was the one. This one is there. This one.
Right here.
Yes, this is very good. Yeah. It's a good request position. The pin already.
What do you have there?
Pardon?
What do you have there?
So in the sample we have 100 mils, 100 microliter of the blood and 300 microliter of the DNA RNA shield.
Oh, yes.
That will make total of 400.
That makes a total of 400, yes.
So add 20 microliter
So you-
of protein.
Yes. So we have a separate protocol for storing samples in DNA RNA shield. We should be able to share with you at the end of the day. It's not part of the kit. So but it's normally recommended you use a ratio of 1:3 for biological fluids. So we added 100 and 300 of the DNA RNA shield. Okay.
And you can also pick one, isn't it? To,
One of
I mean 10
Mhm.
to 300, isn't it?
Can I dispose it here?
No.
I think.
Yeah. So I hope you're following the protocol. Okay.
So I
Mhm.
This one comes to 200.
So you can do 10 in 200, yes. Yeah. Do 10 in 200.
So I've already put, I'm supposed to vote it.
So you vortex,
Yes.
vortex.
It's here, right?
No. The next one. Let me show you. Just a minute.
This one. Oh, no, already. We have 70% at all.
Yes, we do.
I need to put
So you can do touch. Should be on.
because I was trying to put my hand and I'm not seeing it.
Sample, blood sample, the added just
So you just use your judgment for that.
So we say probably this 400 to get. This total sample.
Okay.
We are talking 100 from sample blood.
Okay.
We added 300. Because
So remember, when you're doing it, I should be able to judge you at the end of it. You should be able to get something about 20 nanograms per microliter of the DNA.
At room temperature. So
At room temperature, so yeah, so you just, let me give you a rack. Okay. Should be
mine is MW2. Sorry. Oh, you can start.
So you just have
20, so you set for 20 minutes. Huh?
No, that one. This one is already 400.
Excuse me.
Go inside. Don't take the sample of this is one. This one is big. Done. Don't take the sample of
So you work inside the folder. Make sure you.
This is IM or 1M?
That's IM. IM. Huh? So you give me. I hope you captured your numbers. Huh?
Yes.
Oh, it's okay. It's okay. You just pick it from there. Okay.
IM 13.
IM 13. That's your hammer.
AMEN.
AMEN.
One body. Now, how much we have?
Yes.
One volume we added. You transfer it to the speaker.
Okay. So one volume.
One volume is the same one volume.
So one volume is just the same equal volume. But
if you have 100, you are going to add 100. But are we using this one? You'll be having.
No. Yes, so we'll at some point we'll go back to
step number 3.
Step number 3.
Similar to this one.
Yes.
And then you go to 4, which is equal to the whole thing.
Yeah. So we have. So it says continue from step number 3 of the biological phase. Yeah.
Even that math would shake.
Okay. Then you have for 20, then you leave it at for 20 minutes.
For 20 minutes, yeah. So I hope you set your time.
Yes, yes, I did.
Okay. Excuse me. Can I go with this?
Yeah. Why are you using whole blood and not maybe serum?
Okay. So so for this assay, specifically we got the samples from the client with whole blood. Yeah, so we just use the whole blood to do the extraction. So we are interested in the DNA, the nucleic. Yes. So the next step, you wait for 20 minutes at room temperature. So I hope you set your time, because they have different times. Okay.
You're ready to go? 15 seconds.
She said, oh, they know, they literally know. Like, they literally know. Yeah, yeah.
Yeah, potentially.
So I time myself.
Yeah, you can
Mm. 2 seconds, huh? So if you need more of that, I can give you this. Yeah, just use this one. Yeah, any question?
Okay.
Yes, yes. Okay.
So the 20 minutes to allow for the prioritization
To digest proteins. Okay.
So this blood, it's just blood, nothing added.
It's blood and DNA RNA shield. So the DNA RNA shield prevents, it kills all other microorganisms that are in that sample. So it's like a preservative for that sample. Yeah. But it also has some lysis action. Okay, that's good.
So that's the protein.
Yes. So 20 years. Okay. So you vortex, then you
The number?
so you give me your name. Number is T3, yeah? Your name? Kosa. Okay.
No. CO.
Oh, C.
O N
Mhm.
S T
ST.
H
H. A.
N
T.
T I N.
Oh, constant. Okay. Number 6, huh? Okay. Yours is, so you're digesting. Did you set your timer to?
Yeah.
Okay. Okay. Yeah, so the DNA RNA shield also has some lysis properties. Huh?
Yeah.
So, yeah, apart from just preserving the sample.
How long does it preserve the sample for?
Yeah, so you can preserve the sample for up to 30 days in DNA RNA shield.
Yesterday she said it is here like it's 2 years. And if it's RNA, it's 30 days, 1 month.
Yeah.
That's what I remember.
Maximum.
But I think the one for DNA shield
Mhm.
from yesterday was not from Inqaba.
No,
Okay.
it was Zymo.
Really?
It is Zymo.
It was River.
Oh, okay.
Yeah.
That's
River.
So you've vortex, huh?
Yeah.
15 seconds. Just make sure it's properly mixed. Then you incubate. Incubate it at
This one. This one is not just blood, it's blood in the
so you set your timer to no, it's okay. Set your timer 20 minutes. Yeah.
You put an equal volume of the binding
Between that, yeah. I'm not sure. Yeah. Oh, you did some Zymo products yesterday.
River. Yeah, River had.
Okay. That's nice. Okay.
Do we need to check that just the pH? No. Will it matter?
I don't think
Since the pH is
it,
optimized.
it's already optimized, so you don't need to optimize anything. Yeah. Yeah. So I think that's the beauty of science is to make things simple for them. Done everything for you. Yeah.
So you're doing located there?
So I think everyone has their sample. Huh? Okay.
The first person. I guess that was the last person.
Yeah, so we normally store our DNA RNA shield at -20. So we make sure all the reagents are stored the proper temperatures. They're normally indicated on the tube. Yeah. Okay. Any questions so far?
So you do extraction for people.
Yes.
For samples coming to our labs. Yes. Yeah. Exactly. So we, of course, we have a sample reception policy. Yeah. So any infectious sample must come in DNA RNA. That's number one. Number two, we need. We don't really get samples that are that contagious. Normally, we get samples such as sponges or something. Yeah? Environmental samples. So there's not really much to do in terms of preparation of the sample before it's being processed for it. Yeah. But this is a special case. And then again, I decided to use these the isolates. Yeah. Exactly. Yeah.
So is it you have like a policy that you only want environmental, like you don't want like infectious samples, or it just get customers who?
So if we get all kinds of customers. So if you can process your sample from the lab, then bring the DNA here, sequence your samples here. But as of now, we still have that policy for non-infectious samples. Yeah. Yes. For the protocol. Okay.
You put an equal volume of the binding.
So you always make sure you read the next step before you. Sometimes people forget to do the incubations. I mean, to put on the heat block on time. Eh? So they mess up the whole assay. Should be yeah. Should be three.
You say
I don't know. 100, 300, 420. So you put 420. Yeah, 420. Yeah.
I think it's the big package. That's the first one. So you have to say, yeah.
Question is the procedure. So wait for that time.
Okay.
Tell us now for 20 minutes. What next?
Okay. Okay. So you said you're doing your PhD at that's okay. Okay. Okay.
Right now.
You guys
Okay.
Thank you.
You should be able to do it. Yeah, we do it. Yes, fine. Yeah.
Because I want to do that genetic tree.
Okay.
So I can understand the strain.
Okay. So that's from which isolates and which?
The bacteria is an agrobacterium.
Okay.
It's found in
Okay. Okay. Yeah.
able to produce what you call cell.
Okay.
But I don't interested in a lot of fatty acids. That's the same something. To the morning. But this one is like toxic.
Okay. So
How much would you charge me for that?
so
Now that I'm wearing the laptops.
yeah, so it depends on the number of samples you have. Yeah, so if you do, you have 5 samples. Okay. So that's 5 times 2 it should be times 850. 5 times 2 times 850. Okay. 2 because we do the forward and the reverse, then 850. Yeah. So per read we charge 850. So it's basically, Sanger sequences 850
Okay.
bases. Charge per base for that. Yeah. Yes. Or you can also find the protocol in fonts if you. Yeah. It's readily available online. Yes.
For example, I have my bacteria and some plate cultures. So what's the procedure if I want to extract? Do I have to again culture it in a broth or something like that?
So you have different isolates in the same plate.
Yeah.
Do you have background in microbiology?
Yes.
Okay. Yeah, so I think it would be a bit of a problem with the Sanger analysis because we'll have mixed species in the profiles.
So let me say I have the pure culture.
Yes.
So I want to do the extraction.
Yeah.
So what's the procedure?
So for pure cultures, we also use the same protocol.
Yeah.
We use the same, I don't know where the protocol. So you just follow. So this kit is a bit versatile. It does many samples. So it suits many samples.
Yes.
So you get the protocol for, I think, biological fluids. So you can suspend your cells in TE buffer. Then you can use the same protocol, the biological fluids protocol.
So we need to use the same amount that I would want, maybe the volume that is required to start, or is there some requirement for the volume that you need to start with? Or you can just swap it.
Yeah, so of course there's a volume.
Yeah.
So for the suspended cells, you normally take 100 microliter. Yes.
So you have to get it suspended
in TE buffer. Yes. Yeah.
So the
So I think I forgot to say, during sample reception, we only accept pure cultures.
Yes. That is why it's supposed to do some
pure cultures. If you're doing bacterial identification, we only take pure cultures, the clean colonies that are pure. So we can't do Sanger analysis with mixed cultures. Yeah.
So what if if you try something, I treat the cancer cells with an extract, right?
Okay.
And then I need to accept the RNA and do an RNA sequencing. Yeah?
Mm-hmm.
So how should I bring you? Am I going to bring you the culture itself after treatment? I'm going to give you the RNA itself.
So you can bring the cultures. You can also bring the RNA extracts.
Because the culture might be hard to transfer 4 degrees with
Correct.
5% CO2.
Exactly. Yeah.
So we can bring you the RNA extract.
You can bring the
Do you
RNA
have
extract.
conditions for the way of extraction, conditions?
I mean, we don't really restrict the client on how to extract these samples. As long as the samples come here, I mean, you clearly label your samples. If it's RNA, it's RNA. So you don't really have restrictions on how to do your extractions from your end. We'll accept your sample.
But
Yes.
you must clarify the sample.
Exactly. You must specify it's DNA, RNA. Yes.
And you do quality control before proceeding like sequencing instead of
Yes, we do. Yes. So we do for DNA we do nanodrop quantification. We don't have qubit, but it's something that we get in fusion. But yeah.
So it's only the concentration in terms of concentration.
Yes. But there's also a way you can optimize your gels to do your quantification, as opposed to buying the qubit, which uses reagents and you need to. Yeah, it's a bit expensive to maintain. Sorry. Yeah.
I'm still interested on the bacterial aspect because I know it will cover something. One that can be back.
It's okay.
To do something.
It's okay.
So you say that if I have the pure,
Sorry, just a minute. So you can use the new one.
The first one here, so there are some people who will use the new one.
You can.
My result, you know, Brian is getting angry. No, trust me.
Let me tell you now that if you have the contamination, you have a solar responsibility. You have a way to open them.
I think we know the new one.
Yeah. If we have contamination, it's you.
It's okay. It's okay. So
There
you,
was so much. Yeah.
No, it's your own ways of handling the sample.
No, it's everyone. We will know. Don't worry.
so you can just sit. Let me open this for you. So just finish. So you can put your legs in.
So I'm supposed to use this one.
So you're
Because I've
supposed
already
to,
put the
you
buffer.
transfer. So first of all, you need to label your tubes before you,
Yeah.
so just put it down before you do that. So you label the yellow one.
The yellow one.
So this one you throw it away.
Which pipette? Which tip? This one. Oh, I'm not supposed to be.
Okay. Mm. So you can use this rock. Okay, sorry. So we will wait for everyone to
Okay.
begin this. centrifuge together. Yes.
So,
It's always good to, yeah.
so that way.
So that's one volumes. So one volume is basically the
Where they are
same
saying you to put in new
in new column. Oh yeah.
there's no
So you've done step 3.
no not yet. They still have any.
You still have 5 minutes. So I'll detect these tubes. I think everyone should be labeling the tubes.
We add and then
Yeah,
that's
that's the
Yeah. Sorry. You can you pass me the screen columns? I mean the collection tubes. So you pick one of these, or you never used it. Okay. Then you pick one of these. Pick one. Oh, you want to transfer?
Yeah.
You have them. You label them with your number
So you label them at the top.
at the top only, because you throw the one at the bottom. So you just label once. Oh, yeah.
So you put them together first.
Then you label at the top.
Then you put the
So you transfer the whole. So you've added the equal volume of that genomic binding buffer
then
you
after
add
in the code. Yes.
yeah.
Then you transfer everything. Yes, should be about 850 microliter.
Then you wait.
Then you wait. I mean, you spin it down. So you spin it together.
With this whole structure together.
Yes. So that once you spin, sorry. When you're spinning, the waste will come at the bottom. Then you remain with your bound DNA.
DNA bound to this. So next is to mix this.
So you mix that again.
Oh.
So exactly.
So after
you
adding
put,
that one volume,
put these at the top, then transfer everything. So you need to you always need to work in there. Or the markup pen.
mark up.
Okay.
So we need to centrifuge at this.
Yes, so
valid
this
for 1 minute.
for 1 minute.
Oh,
Yes.
so that's
Huh? I should tell you. Okay. Oh, you got it. Yeah. Genomic binding buffer. Genomic. So you follow the protocol to the letter.
yeah.
So it's always good to let me show you just a minute. Let's show you something. Let me show you something before you proceed. That's a new tip.
Yeah.
So it's always good to early quote here so that you don't contaminate the whole bottle. So I think you released very fast. That's not enough.
I wish you opened all this for us.
Up here, yeah.
Right?
I did already carry it.
Just do it because no, don't open. Just leave it closed. Just put it on top. So you can pull the chair. If you release, just pull the chair.
Up and down.
Yeah, so you have to vortex. So just sit. You sit.
I'm holding it. That's why.
Okay. So you've done pipetting before?
Yeah.
So many times. Huh?
Yeah.
Okay. So you pick your tip, then you go to the first stop,
Yeah.
and you pick your sample slowly. Don't aspirate very fast. Okay. So you pick, you put for him.
First go.
Okay. And you dispense. So if you touch the tips of that tube, you have to throw away the tip. Okay.
Which button?
And you close the bottle. Okay.
Which button is
Then you
there
can-
or any
Yes. She's ready. Okay.
I think the I wanted to avoid the traffic.
They can just increase it to pick the whole sample at once. Like,
Yeah, you can do 850. Do 850.
You know, I went to and 6:30.
which one is mine?
So spin it down to make it minutes. Sorry.
You're starting to do?
No, I'm just doing a short spin. I think it overflow a bit. Yeah.
What do you mean overflow? Because I really also want to learn to troubleshoot.
Yeah, so it's always good measure to spin down when you're mixing your buffers, just to, sometimes when you're pipetting you get some bubbles and it spills over, so just
Bubbles.
yeah,
I think also it's for no. You pipette?
pipetting,
Yeah.
yeah.
Sometimes I have.
Yes.
I think you've taken one that has
No, none.
you are spinning.
Yeah,
When it finishes, it's open.
yeah, it does open. It opens automatic. So you can see it's a bit.
Here now.
Yeah, so you can do it. You can work without contamination. As a process. So it's not in the protocol, but you always do it. You can do it for. Okay. Sure.
So that you get the chance.
Yeah, it's always good measure to just. Yeah, yes. Depending on your skills of pipetting. It's okay. It's okay. It's just lysine. So it's nothing. You can do it you can do lysis overnight. You can do it. Yeah. It's always good to sit when you're doing. Yeah.
It could any fact like this. There is an amount of blending back.
I don't think this
No, it won't. It won't, but most likely it will reduce your yield, your DNA yield. Yeah. But it will still capture, I mean, it will still bind your DNA. So always avoid, when you're working with buffers, avoid touching the sample with the tip. Yeah. Just avoid touching the
From the side.
Are you transferring forward? No, just no.
Okay. I'm not still.
Their friend.
Yeah.
No.
That's 850. So I think that's enough. You don't need to put everything. That's enough.
Can I speed it down before?
Yeah, you can, but you have to balance it.
Yeah.
You have to balance it. So there's put a few balances at the side. The other side. Yeah. Okay. So that's your sample.
Jackie, is that right? Empathy?
MW1.
So you make sure you transfer the right sample, the correct sample to the correct tube.
MV13.
Okay, that's enough. Don't introduce bubbles. Once you transfer, you discard. You can discard. Discard it. So you always want to avoid this kind of mess.
Yeah.
Yeah. So I think if you put it up to here, that's enough. Just avoid the bubbles. Yeah.
It's already set.
So so make sure you put it in the correct tube.
The amount of water from the start.
From the start. Yeah. These are Myra. It's an auto. It's a liquid handler. You can do your extractions. You can do your library preps. It's a bit versatile. So avoid bubbles. Just pick the bubbles before you throw out the tip. Just pick the bubbles at the top. Yeah. So you always, before you go into the sample, make sure you go to the pass stop before you go to the sample.
Okay. That's why.
Pardon?
They put 850. That's why
It's overflowing. Yeah. But you don't need to put everything. I think 700 is enough for that. Okay, that's enough. So if you close it, you won't get these that kind of. Okay. It's already set to 850. So you've added the genomic binding buffer?
Yeah.
Okay. How much?
420.
420. That's it. Okay. So yes. Okay. Yeah, that's good. Guys, so let's deploy the chip. Let's go. Mm. Yeah, we many sections. Yeah. So yeah, we have the plans. We do have the plans. Yeah. Exactly. Mm, yes, that should be just add a bit. Ongeza tu kidogo. Yeah, that's enough. That's enough. Okay, I think we have 8 samples. Huh? So we spin. The next step on the protocol is to spin.
Just say the volume.
You can just check. You can check visually. Just check it. That's enough. Yeah. Yeah. So you carry the whole batch. Let's carry the whole. Oh, yeah. Yes. Yeah. So you. So it should be 800. Yours is a bit small, but it's okay. It's okay. So what's the next step? Spin for 1 minute. Huh?
Is it a cold spin?
No, it's not refrigerated. It's a normal one. We normally use cold spin for preparing plates for sequencing. Yeah. So can you set the time? Yeah, you set the time.
1 minute.
1 minute.
Set the speed and time.
1 minute of time.
So RCF is also similar to G. Yeah. Yeah. So you start.
You also RPM.
Yeah.
Yeah, we have.
But it will be different according to the size of the axe. The rod.
Yes, yes, yes. Okay. So you can change your gloves if you feel change. Yeah, maybe after tea. After, but we have to.
We need to include.
Yeah, we need to elite. We need to elite before we go. Yes. Any questions so far?
Yeah. My question is, okay, we are extracting DNA, but what is the sample? What was the sample? What is our main goal in the particular?
Yeah. So as I said before, we want to do species ID, but we get difficult samples like plates. So we don't have a proper clean plate for that. So I'll do a demo for extraction using the blood samples. Then we proceed to PCR with species ID all the way to sequencing. Yeah, but we won't use these samples for species ID. Yes.
We are doing species ID.
I already have DNA for species. Yeah. Yes. This is just to take you through DNA extraction, but it also yields more DNA. Yes. So I used Bacillus BT. Yeah. So we had some cultures here for BT. Then I extracted the samples last from bacteria. Yes. Okay. Gloves. The gloves. So you pick the samples.
Yes, we need.
Yes. What she can then. Yeah. It's out. Okay. So I don't know.
Can I come check?
Yeah.
So we complete, then we go back here. So that number 4, so we go.
Washing, right.
So if you're hungry, you can go for tea. Then I can, you can come back and do the washes. Or I can do it for you. You need to do it. Okay.
I need the
Sorry.
correction. No, I want so that the time I'm moving, I'm pushing it around. I want to go.
Then I think we just do it quickly before we go for tea. Just wash. Yes. So you throw away the.
We discard.
Yeah, you discard the spin cord. I mean the collection tube. Then you get a new one, fresh one.
I don't think I need a
It's already.
Yeah.
So you throw it.
Discarded.
Yeah, discard it. Then you get a new one, collection tube. So you start with the pre-wash. Pre-wash, eh? Yeah. 400. So you early coat. So make sure before you touch the sample, make sure you early coat your buffers to prevent any contamination.
So it's better I put it here.
Yes. Yeah. Anytime you're working with buffers, make sure you do early coating before you touch your samples.
Then what? Yes.
So no pressure, just take your time. Yes. So we do it fast so that the same thing. So we get a new one, this one, new collection. Yeah. Collection tube. Then let's pick one.
If they accept, I'll do it. All of them necessary.
Yeah, you can do all of them.
Okay. Okay. Nice. That's
So you can throw them here.
you have for it here. Behind you here. Please change the
Okay.
give me three collection please. Collection please. Do you have the collection?
Yeah.
No, but the collection is don't take that.
Yeah. So you always want to avoid contamination. Just make sure you sit down. When you're working in the hood. So I think I've seen some blood in that sample.
It has not been transferred.
So you need to spin it more.
It's from down. The membrane is clean.
The membrane is clean.
Yeah.
It's okay.
Okay.
Okay. So remember, I gave you a simple sample. It's a blood sample. You expect to get a lot of DNA. So if you don't get it, it's definitely a mistake. Yeah. So don't pick directly from the tube. Always a liquid. Otherwise, you'll contaminate the whole batch. And yes. Then throw away the tip. I think you touched the tube. So if you touch a tube, you throw away the tip. You add some more there. No, it's not enough. Yeah. So you need to add some more. It's not enough. Okay.
I can return?
Yes.
Yeah, just return.
Yeah?
Yeah, it's okay.
Just for that.
Yeah, so
We sent it to you.
as long as you've not contaminated my buffers.
Yeah, that's the other thing.
Yeah. Okay. So do it for another minute.
Someone can go to the next.
Someone can do the other wash. Next wash. So you can just do 13, 14, 15, 16, then 1, 2, 3, 4. So there are 8 samples. It's okay. It's okay. So, unless, so you make sure the cups are closed, all the cups.
We also discussed.
Yeah.
Then
Okay.
you
And you spin
Another
it.
one for
So it's like a click fit. Yeah. So you can Unaweza ukanini? You can, excuse me. No, some blood, some whatever, some sample there.
Oh, yeah. There.
So.
No.
Yeah, but it's infection free. Yeah, so it's there. Yeah.
They should just do it slowly because one shit there.
Okay.
Like it has been used in a different location. It's down. Customer is at the 700 and we are going to put that enter.
No, no, it doesn't answer.
Now it's 700.
So you can throw them away, but I can also show you another way to conserve on
You
this.
empty it. Another
Yeah,
way you empty,
yeah. Then you tap on pepper
So
towel.
what should I do?
Just throw them away because it's,
These are one use. You can't recycle.
sometimes you get like a few. Yeah. You get like you've used most of them, so you don't have enough. So yeah.
And then like if you use it for the same sample and you are just doing washing,
It's just washing. There's no effect on it.
you are not irritating on the phone.
So so you open first.
Yes. Is it
Open.
okay if I open all of them?
Just open all of them. Aliquotius buffer. So I want to see if you can use a single tip for
Actually,
all
I
the
will
samples.
use a single tip.
So you also try to save on your consumables when you're working in the lab then. Okay.
No, it didn't. It did not.
Yeah. A bit, just a bit. So you can squat it away from the tube and just use enough pressure. Up or then just push. Yeah, you go. That's so you throw away the tube and you shake the sides of that. Yeah. No pressure, no pressure.
Be careful of that bottom you don't.
So the filter tips are much more expensive than the non-filter tips. So you always try to conserve them.
No, I didn't mind it. It's a normal thing.
And
You're supposed to
yeah, for holy,
you recommend actually.
not water. Yeah, it works. Yeah.
I'm wasting a lot.
Yeah.
You have it.
Mm.
Wow.
So now this buffers, see, could be using a lot of it. That's why I was talking of reducing the amount, the volumes, the volumes.
Okay.
The buffer comes with kit.
So the kit is meant for 50 preparations. Yeah, so you just the buffers, yeah. So you use the buffer is meant for exactly 50, maybe plus 1 or 2.
And you know the way company is doing it. If they do it, I think the 50 is 50.
They can't. Yeah.
That's all the company. That's all the company.
I don't think
Especially
it
if it's one year.
because you know when
They do big things. Because I look at end up in the 50, end up in 40.
So you confirm that all the samples have the buffer. Just confirm.
it's hard pressure. You make me shake. I don't usually shake when I'm pipetting. Like pipetting is usually my thing.
So you just ensure they're balanced, then you close with the cup. The other cup. Otherwise the tube will just fly inside there. Just fly.
There is disparity in the terms of volume. Why is this one?
So I think that the tube can hold a maximum of 1500. So they just split it into two.
Okay.
Yeah. So if you were to put 900, it will overflow. So you just split it. Yeah.
This one is way better for 200. Maybe prefer this one. Compared to that one. Let's find out.
200? Can we reuse these? Thank you.
So the last wash, the final wash.
Yeah.
So you pick the pipette that is closest to your volume. The pipette that is closest to your volume. So for 200, just use 200. 200. Yeah. 200.
So example, for example, tissues could be over observation. That's the ones for histology.
Solid tissue, yeah. Yeah, we do. We also get sponge samples. Sponges, huh? The ones from the sea.
From the marine.
Yeah, marine. Exactly. So you can use the protocol to do that extraction. I could have given you that, but it's a bit difficult to use it. Yeah, to get the DNA, it's a bit you have to modify the protocol.
We have another project.
Just we have. Let me bring.
What's the volume of that?
So you can pick 200.
Oh,
So throw away the tip. Put the tip.
no, just discard the one that you put.
So that's 200, eh? Okay. Okay, that's better. That's better.
Without touching?
So just just away from the tube. You conserve the tips. Yeah. So you close your buffer before you start. Yeah. The point is just avoid contamination. Otherwise, if you contaminate your buffers, you'll never get results for your assays.
How frequently do you do the aeration?
So we do it daily. Daily. We do it every day. Daily. Okay.
No, now you can. Because you're going to eat. Between the washings you could have, you could have it. You don't get one. But now, you can get in there. You have it.
Appendix. So you need to start labeling the appendix. Yeah. So you pick, your numbers, you label them. Okay. Then So there's a pre-wash, then there's a wash, then there's
Yeah, we watched only.
I think it should be just ethanol for all the washes. Mostly ethanol and just salt. Yeah.
Just in the matter. What percentage do you normally use for washing?
I mean for the
Let me say
buffers.
you're doing a crude extraction.
Okay.
Yeah. So what percentage?
I can't even remember honestly. Yeah, because I'm used to using kits, so yeah, I can't really remember how much.
And does the kit fall under mechanical, chemical?
It should be chemical. It's chemical. Yeah. So I put my illusion buffer on the heat blocker. Yes.
Okay.
Yeah. So
It has to be.
so you can use it the way it is, but for maximum yield, it's recommended to preheat it.
Other than nucleus free water, because we can edit it nucleus free water, but it depends again on the case. What are the other types of emission buffer, except like within nucleus free water?
Okay. So for sequencing, especially NGS, you don't elute with trees HCl.
Yeah.
So you do your elutions with either water or TE. Yeah. So there are protocols we have here for elution. It's just 3 CDT, the pH of 6 to 7. Yeah. Yeah, so most sequencing labs use water to do the elutions. Yeah. I also use water, but I just wanted to use the kit. So, yeah.
So it means you follow the protocol, but when you get here, you
I use water. Yeah. I have a little of water.
Okay.
Sorry. Okay. So I have water, nucleus free water for all my assays.
Imagine that we are after the shipping nucleus free one.
Yeah, you need to start your own production.
What now? If you don't water,
Yeah, either. Yeah. Okay. So you make sure the correct tube goes to the spin column goes to the correct tube.
when you're touching this thing, touch here. Avoid touching the upper part. Okay, that's
Will be touching.
Yeah, there we are.
Oh, sorry.
Actually, don't know.
In the 13th, so you need to write.
Who's the little? I'm gonna joke.
So you don't, I don't normally alute from the hood. So you do it at the bench outside.
This centrifuge for maximum speed for 1 minute, which means what maximum speed? Is it 14, 1200 or 14?
So you can normally I do 14, but you can go with the maximum. I mean, there's preset speed on the centrifuge. So you can use the 200. 200. So you set it to 60.
DNA emission.
Where is it?
Yes, so you can yes, open this. Open this elution buffer.
I got it from here.
Yeah, just open it. You can put the pipette down, then open it.
Which volume?
And aliquot, you can aliquot. Just pour it on there. Okay. Don't shake, just put it here. Yeah. That's enough. That's enough. So you can put it back here. Then you can bring your samples here. So you open all your samples first. Sorry. Just make sure you don't. Yeah. Just open them properly. Yeah. Okay. Yeah. Okay.
No indication of expiry.
Yeah. So so most of the buffers don't really have expiry dates, but yeah, it's just most of it is just ethanol. Oh, sorry. So make sure you put it on the matrix, on the white thing. There's a white thing in there. So, okay. So that's 16. Make sure you put it on the matrix. You don't have to. Let me show you. Just show you. Show you. So you put this and this. Okay. So you pick, just pick 160. Then you drop it on the matrix. You don't have to go in. Just make sure you just put it on the white thing inside there. Just like that. So you can do the rest. So it's always good when you're working with many samples.
Yeah.
Once you put the lesion buffer, you close it. Otherwise, you just confuse with it. You won't know until the end. So you just. Pardon?
Yeah, outside
Yeah. So so that you can see the matrix clearly. It's always good to do it. I normally do it outside. Yeah, but you can also do it in the hood if you prefer it.
now, especially if in those when you put when you come to it in an open dock, it cuts
The cups.
how do you do it?
So there's a way. I'll show you how to arrange them. So if you missed one, you won't know until the end. So it's always good to, once you put the illusion buffer, you close it. Yeah. So this way you can't even, you can't know. So incubate, so you set time for.
Just collected.
The sound you get it.
Yeah, collected. question.
Yes.
The temperature.
So the temperature is, I put it at 65, just for maximum elution. But you can also use it at room temperature, but not cold.
I have a question.
Yes.
I have a thermoficial kit where the first step of lysine,
Mhm.
it needs a preheat of the reaction of the reagents to 65. And the incubation generalizes at 54. And it's an exogenous RNA. Wouldn't that degrade the RNA? Because hitting at 65 is the reagent, and then the incubation for 10 minutes, it's already in a water bath of 65. Wouldn't that
Degrade the
the RNA?
Okay. So was the kit meant for RNA or?
The kit is already meant for RNA. That was the protocol.
Okay.
And then I got my
Okay.
like, yeah.
Mm.
With the nano drop concentration,
Yes, yes.
the quantity is there. But when you go to the PCR, nothing is amplified. So the RNA is there, but like kind of degraded or something like that. We did not do a gene electrophoresis. But now we suspected the high temperature because you know RNA is even more sensitive than DNA.
Okay. So you're working with the RNA on gels? Did you have to do some cDNA prep?
Yes, after extraction, we have done this.
So I won't suspect the kit. Mostly I just suspect your protocol for your cDNA process, preparation. Yeah, so probably you need to change the protocol for
So even
cDNA.
working with a kit that requires high temperature will not affect
It won't
the
affect.
quality of
As long as the protocol says you use yeah, it won't affect it. So you need to maybe try a different cDNA preparation procedure, yeah, because they're really difficult to get to cDNA. Yes, yes.
Oh, the time is up.
It's okay.
Now I want to see how we put them.
Oh yeah, so I normally put them this way.
Or you put it down.
So put it this way.
Okay.
Yeah. So if you have even 30 of them,
Okay. To not with that.
they won't break. Yeah. Yes.
Another paper that I always keep them on the upper side.
And then they'll break.
Okay.
They'll break. Okay. Okay. Okay, so make sure they're balanced. So maximum. So normal. So I normally use 14. Anything around 14, just give them 1400, yes. Then we can go for T and come back and do quantification. Hmm? Mm. Ishapoa. That's bad. Umefika mapema. So you can use the other lab here. There's soap on the other side. There's soap and everything. Yeah, you can just leave them on the seats. So just find a seat, like a lab 1, lab 2, just feel free. Just put them on the seats.